Hey there, microbiology friends. So this is the lecture I probably should have done yesterday, but I got caught up with other things and I'm behind. What else is new? So we're going to continue talking about cells, and we were going through the structures of different cells, cell membranes, and the components of cells, things like ribosomes, the cytoplasm. I'm looking at myself in the video monitor. I have to trim my beard. Oh well. DNA, and we finished up with the idea of what we call the central dogma, the notion that DNA replicates and DNA is transcribed into RNA. RNA is translated into proteins. So that's for the basic cells. Now, we've been talking a little bit about comparing the cells that don't have nuclei, the prokaryotes, with the cells that do have nuclei, the eukaryotes. You are a eukaryote. Now, what we're going to do is shift gears a bit and actually begin talking about bacteria and the prokaryotes. So, we can break prokaryotes down into two different types of classes. And one of the things we're going to be thinking about here is how we can begin identifying bacteria and the idea of species and what are some of the characteristics that we look at to identify and distinguish different species, and something that we call a dichotomous key, which we'll be working on a little bit as we start kind of doing a synthetic lab. A dichotomous key is a tool that allows you to have sort of yes-no type questions that lead you down a set of branched decision points, and you finally get to a spot where you go, okay, if you said these answers to these questions and it's led you down this tree, you end up at this point and you say, okay, it is this kind of bacteria, it is this species. So that's what we're going to be kind of thinking about, are some of the standard observations that are used for a dichotomous key and how we can begin thinking about that. And we're going to be doing, again, kind of a mix of PowerPoint and board discussion. But just to start with, let's start working a little bit on thinking about the two ways of considering bacteria or prokaryotes. And this is a bit of a new idea in the past 15 or 20 years. The idea that previously we had prokaryotes, and they are all the bacteria, all these organisms that don't have cell walls, or they don't have nuclei. Now, once people began studying and actually identifying more diverse species, it became obvious that there actually is a subtle difference within this general group. And so we have two different types of prokaryotes. Type 1 is what you would typically think of is a bacteria. And for our purposes, the two different types are very, very similar. Type 1 is bacteria. Type 2 are a class that are known as the archaea. Oh, I started getting ahead of myself in my writing. Type number 2 are the archaea, or the archaebacteria. And these are the ancient prokaryotes. These are the more evolutionarily distant bacteria. Now, let's go ahead and, just for the heck of it, turn some slides on and look at a few things. And a couple of good definitions come up. And you have these PowerPoint slides that you can look at as well. So you got to kick up a little bit. There we go. And wait for it. Wait for it, here they come. So we've got the prokaryotes. And here it comes. Pretty good contrast. There we are. All right, so this is the type 1. And we refer to them as bacteria. Now, bacteria as a group of organisms have a variety of different kinds of sizes here and shapes. We have these long rods called bacillus or bacilli. We have spirally guys, spirillum. We have little dots that are staphylococcus. We have the little strings that are the streptococci. And these are a very diverse group of organisms. And one of the things we can say about bacteria is that pretty much anywhere on the planet you look, you'll find bacteria. There is no place on the planet that you won't find bacteria present. So you can go to the top of Mount Everest, you can go into the middle of the Gobi Desert and take a little bit of a sample, plate it out, and you'll probably find some bacteria here of a variety of different types. And we can broadly classify bacteria based on how they get their energy. And the two different types that we consider are what are known as the autotrophs and the heterotrophs. Autotrophs are really weird kind of organisms compared to us because they don't use a carbon source as their energy source. They're pulling carbon in from the atmosphere in different ways for things, but in terms of energy, they're not eating stuff like sugars or fats or proteins. They are getting energy from non-organic sources. Now, we are heterotrophs, and there are classes of bacteria that are also heterotrophs, and they use a carbon-based energy system. Sugar. They eat protein. They eat the flesh on your skin. All right. Now, interestingly, there really are only a small number of bacteria that are responsible for the bulk of infectious diseases. So, medical microbiology is a way smaller field than general microbiology. So, we've got the medically relevant bacteria. Really, it's a fairly small number. So we were looking at the prokaryotic cell in one of those figures earlier. That's a standard bacterium. Now, when we shift and look at the second type of prokaryotes, these are the archaebacteria, or the archaic, old bacteria. They are independent living organisms. They don't need to have cells that they actually interact with. They are pretty much mostly autotrophic. They don't use organic compounds. And they are growing primarily in really, really hostile environments. And that's part of what makes them so very interesting, but also some of the reasons why they are quite distinct and quite different from the bacteria. And these guys, they live in, here, let me get my little laser pointer. Beep, 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 beep. Thank you. They can live with a lot of heat. There are thermophiles. These are bacteria that live in near boiling water. There are other organisms that can live in not boiling water, but near very high temperatures above 110, 120. We can't actually survive. There really aren't very many eukaryotic cells that can survive at that kind of temperature. There are bacteria that live under really high salt conditions, known as halophiles. They're living in seawater, but they're also living in very, very salty conditions that can be found like in the Great Salt Lake and other places on the globe where we've got the Dead Sea, for example, where you have extremely high levels of salt. There are methane-producing bacteria known as methanogens, and these are a fairly weird, odd class as well. There are bacteria that can grow in acid. There are mining operations that generate large amounts of acid tailings. They actually have acids floating around in the refuse from mining operations, and they have these ponds they hold this acid in, and it is a phenomenally toxic environment, and there are bacteria there. So they are a really interesting class of bacteria. Just as a little note, there is a primary diagnostic tool being used these days for the analysis of the COVID virus, known as the polymerase chain reaction, also known as PCR. And it requires the use of an enzyme that can operate or survive boiling conditions. And it turns out that there is a bacterium called Thermophilus aquaticus that was identified in a boiling water event like in Yellowstone National Park. It has a group of proteins that are able to live and actually thrive in near boiling water. And so the Thermophilus aquaticus DNA polymerase is a critical tool in the detection of COVID-19, and that's actually a part of a process of PCR that is a critical tool for pretty much modern molecular biology. And it has an operating temperature. Its best sort of conditions are around 72 degrees centigrade, which is really, really hot. The boiling being 100. It can withstand boiling temperatures for probably 30 or 40 minutes without being denatured. That's rather remarkable. There are no proteins in our bodies that can do that. So there's a whole range of different kinds of bacteria that are actually quite interesting, quite useful. On the other hand, if you look here down at the bottom, no human pathogens. So, truly, we're not going to be talking about the archaea much anymore for the rest of the semester, although I find them quite fascinating. So that's, in terms of microbiology, we don't need to worry about those guys. What we want to do now is focus on the identification of bacteria, particularly pertaining to those kinds of bacteria that are disease-causing. Now, this is an evolutionary scheme of life that we can develop looking at organisms that give rise to infectious disease. So it's not inclusive here because you don't see animals much up here at all, but few that give rise to disease. You don't see peoples up there. But this is a way of beginning to split apart living organisms based on specific characteristics. And it is a map of the way organisms have evolved over the note of the billions of years of time here. There are always changes being made to these relationships. One of the things that is a modern tool of understanding this is the use of DNA technology. And as we sequence more genomes, we begin to see more and more relationships, and it changes the overall relationship. But this is a pretty good map, okay? So we've got a whole range of bacteria going on this side. And we have another set of organisms known as the protists. These are a variety of different kinds of single-celled organisms. And then as we continue up here, we get into the multicellular organisms, things like plants and fungi and animals. Here are the archaebacteria. They kind of have a separate branch of this tree. Now, what's here? This is some of the progenitors of the earliest cells that gave rise to life on the planet. We really don't have much information about what those are. But we can begin to separate these things out. So we've got a whole range of different bacteria types, relatedness. It's kind of like a family tree. And there are groups of bacteria that have similar kinds of characteristics, are involved in a variety of different kinds of diseases. The proteobacteria, these are a whole range of gram-negative bacteria. We'll talk about staining here in just a bit. They are responsible for a lot of pathogenic bacteria, things like E. coli found in your gut, but the bacteria that cause plague, dysentery, a variety of different kinds of diseases. Spirochetes are another kind of bacteria, sort of an oddball. They, members of the spirochete family, are responsible for Lyme disease. And we've got a whole bunch of other guys that are part of here, like staph, strep, botulism, lots of different kinds of bacteria can be placed over in this range. And then there's a whole set of bacteria here, photosynthetic bacteria, that are probably the source of chloroplasts in plants, and a whole range of bacteria that are not pathogenic and not involved in human disease at all. Now on this side, we have things that really are not so much part of our specific class, our course, in that we've got amoeba and slime molds. These give rise to dysentery. We've got a whole range of different kinds of protists. These would be things that you might see in pond water, like you've seen paramecium or amoeba and things like this. And these also give rise to specific diseases, things like trichinosis, and malaria is over here, giardiosis, leishmaniasis. These are various sorts of tropical diseases often. Then we've got in the fungi, this is where yeast are present, and yeast gives rise to things like thrush, and you can have yeast infections in various parts of your anatomy. You can have infections, fungal infections on the skin that result in athlete's foot, jock itch, things like that. Then we've got animals, and there are a few animals that actually give rise to relevant medical diseases. Things like flukes and worms, leeches. We don't have to worry about that so much. But ticks, while they are a problem in various insects, they are animals, but the tick is in fact the carrier of the spirochete that causes Lyme disease. So those are some things to think about. Now, one of the ways that is an interesting notion with the cell business is this idea of evolution using bacteria as a way of going from a prokaryote into a eukaryote. And this is something known as the endosymbiotic theory. And it's a little point we want to bring up, in that we carry organelles in our cells that actually are very reminiscent of bacteria. So every cell in your body has got multiple mitochondria, and mitochondria are actually, they have characteristics similar to bacteria. They have a circular genome. They are generating transcripts and carrying out protein synthesis in a way that is a little bit like the way a bacteria would. It's something interesting to think about, is that, with inside your cell, you've got your nucleus with the chromosomes you get from mom, half, and you get the other half from dad. In the cytoplasm, each cell has 5, 10 dozens of mitochondria. Mitochondria are donated strictly from mom. They are maternally inherited. And they've got their own DNA. They replicate inside the cell. And they are not able to operate free living. In fact, they have a lot of coordination between the nucleus and the mitochondria. But they have some interesting kinds of characteristics that lead to this idea that at one point there was this ancestral prokaryote that began to develop different kinds of enfoldings of plasma membrane, and that became the endoplasmic reticulum, this membrane material that is responsible for membrane biosynthesis. And we end up with cells that have a nucleus as these membranes begin to organize themselves. And then at some point, a bacterium was engulfed. This is a aerobic bacterium, a bacterium that is able to use oxygen. There are bacteria that are known as rickettsia-like bacteria or rickettsia bacteria that only can survive within cells. I mean, these are current. This is something that actually is a current cell right now. People sometimes they have organ transplants can end up with rickettsia infections, but this cell engulfed this kind of bacteria, and this becomes the mitochondria. The mitochondria and different kinds of systems become stable, and then a photosynthetic prokaryote, like a cyanobacterium, gets engulfed, and that becomes the chloroplast. So we end up then with these modern complex cells that have arisen from the very primitive cells, bacteria, that they may have actually encountered. So let's, though, get back and stay with our discussion of the bacteria that are really the most important guys in medicine. And we've got a class of bacteria, and I would say to you, probably writing these down and generating a list of their characteristics would be a useful thing, because this is a class of bacteria or a set of bacteria that we're going to be talking about a lot. So first we have the protobacteria. The protobacteria are a very diverse group, and they have a range of different kinds of bacteria types, and they are giving rise to a variety of different diseases. Food poisoning, Salmonella, Klebsiella is a great one for generating pneumonia. Yersinia is the bacterium responsible for bubonic plague. The Firmicutes, these are another kind of bacteria, have a very rigid cell wall structure, so they're pretty easy to identify. And we have here things that you probably have heard of, like Staphylococcus, causes staph infections, Bacillus, Streptococcus, Clostridium. There is a species of Clostridium called Clostridium botulinum, and that causes botulism. It's also the source of Botox. It actually has a protein that is a really potent inhibitor of protein biosynthesis, and it's able to interact with eukaryotic cells and cause them to, if not die, at least become paralyzed. Spirochetes, as we mentioned before, they've got a really weird sort of spiral shape to them, a coil shape and flagella, and they're causing things like Lyme disease, syphilis, very sexually transmitted diseases. Another sexually transmitted disease is chlamydia. This is a interesting kind of bacteria in that it really can only grow in cells, but it produces a spore that's able to propagate and actually maintain resistance outside. And so chlamydia can be, forms of it are sexually transmitted diseases as well. Actinobacteria are another class that used to be considered fungi. They're fungus-like. They live primarily in the soil. Now, they're not really typically disease-causing agents. You can identify them, though, in soil. But what makes them really interesting is they are often a source of a lot of antibiotics. And, antibiotics are often produced by other bacteria and other microorganisms to kind of ward off and push away any kind of competitors. Cyanobacteria, these used to be known as blue-green algae, but we now know them to be bacteria. They are able to carry out a range of different kinds of metabolisms, things like photosynthesis. They were probably some of the first bacteria that were able to carry out photosynthesis during the early evolution of life on the planet, and actually responsible for shifting our atmosphere over from a largely nitrogen carbon dioxide atmosphere into an atmosphere that has oxygen. And without oxygen, you can't have advanced organisms. They also release a whole range of toxins into waterways. You can have things like a red tide, if you ever hear of this. These are algae blooms, and they can generate toxins that fish can pick up and cause fish kills, and people can consume some of this material and also have problems. But in general, they're not infectious. So these are some of the different groups of bacteria. Now, what we want to begin thinking about is ways that we can begin distinguishing bacteria. And we're going to be getting into this both in the traditional lecture portion and also in our virtual lab idea, which we'll be getting into hopefully next week. We'll start figuring out how we're going to be doing the lab a little bit better. But please do not worry about that. All right. So we've got the problem of identification. And this becomes a difficulty because bacteria, A, are small, and so in order to really visualize them, we've got to look at them with a microscope. And to the casual observer, they look pretty much alike. So how do you identify a pathogen? Say a patient comes in, you do a swab or some kind of culture, and you look for bacteria, and now you have to identify it. Well, as a molecular biologist, there is a modern way of doing it, and that's to crack them open and sequence a bit of their DNA. That always works. That's not always something we can do, and it's not the traditional way. And we need to think a little bit more about some of the ways we can actually begin to identify the bacteria so we can distinguish which ones are pathogenic and which ones we don't have to worry about. Because you're covered with bacteria. Most of them, happily, are non-pathogenic. Occasionally, you may have pathogenic bacteria start to get a foothold in your lungs as part of pneumonia or in a wound. And it's important that we can begin to distinguish them. So, one of the first things that is done in the analysis of, or the identification of a bacterium, is to begin looking at its basic shape and physical characteristic that way. Then we have a whole series of tests that we'll be talking about. So let's just go through this slide a bit, and you can follow along on your PowerPoint. And we've got some terms that are in yellow. These are some of the terms we use to describe the actual shape of the bacteria. So we have the cocci, or the coccus. These are spherical bacteria. We have rod-shaped bacteria that would be known as bacilli. There are curved or spirally bacteria known as spirillum. Let's see. We've got sort of a combination of these, like a streptobacillus, which is a rod, but they're actually attached, and so they form this sort of string. Coccobacillus, these are not quite spherical, not quite rod-shaped, kind of in between. And so they are referred to as coccobacilli. Vibrio, these have a distinct sort of comma-like shape. Let's see. Oh, the Streptococcus, this is now similar to the Streptobacillus, and there's a chain, but it's a chain of dots rather than a chain of rods. And Staphylococcus, this is the dots, but now they're stacked up one on top of each other. And so that's how you can start to combine these different sorts of terms. And then there are a variety of other kinds of bacteria, but in terms of pathogenesis, those are the big guys. Streptomycetes, these are a lot of antibiotic-producing bacteria come from streptomycetes. You may have heard of the antibiotic called streptomycin. And these are filamentous-like. Here are the spirochetes. These are the guys that are actually responsible for Lyme disease. And they're a bit more stretched out than the Sporilia. And what we have here then is a set of characteristics that we can begin to generally apply to bacteria to identify their species, for the identification. All right. So we've got a couple of different tools, or a set of different tools. And this is another one of those things that's probably a pretty good list to write out and to memorize, the different kinds of tests that we can use to actually begin identifying organisms. So, as we just mentioned, we've got shape. Shape is a biggie. Using a microscope, we can determine the bacteria's shape on a bacteria a spread on a glass slide. Then we can begin looking at structures, things like the cell wall. And do they have flagella? A couple of these things, like ribosomes, require RNA sequencing, but it's another way of we can begin analyzing this. Looking at the presence of what's known as an endospore, or the presence of a capsule. And some of these kinds of characteristics are identified using different kinds of stains. So these are dyes that have been discovered over a couple of hundred years that are going to interact with different kinds of structures. Now, another key test is looking at metabolism, because different bacteria have characteristic ways of breaking down different foods. Some are able to convert nutrients, specific nutrients, into byproducts. Some can't. Some can grow on certain sugars. Some can't. And so by looking at different kinds of metabolism abilities, we can begin to classify bacteria. Another classification system is using antibodies to identify specific molecules. If you've ever been to the doctor or in a clinic where they do, I think what's often referred to as a SNAP test, it looks a little bit like a pregnancy test. When you go in and you have the symptoms of strep throat, what they used to do is take the swab, stick it in the back of your throat, do a swab on a plate, let it grow up, and it takes probably 15, 18 hours. Then take a bit of the colony, put it on a microscope, and look at it on the scope to see if it is in fact a streptococcus. That is, a whole series of strings of dots. And they would stain it, and they would make a reasonable judgment about whether you had sufficient numbers of these to qualify as having a streptococcus infection or a strep throat. Now, typically what they would do is, if the doctor's, under his judgment, says, yeah, you probably got strep throat, they'd start you out on antibiotics as you left the office, knowing that it's going to take them at least a day and a half or so to actually do the bacterial culture and the spread. And you would often have people that are on antibiotics that don't have strep throat. And it really is not a very good way to operate. What we have now is an antibody test in which they take the swab, they go in the back room, open up one of their little test kits, snap it, put the swab in there, and we have an antibody against some of the specific proteins and carbohydrates on that bacteria strain. And it lights up positive, or it doesn't light up, it's a negative. And so before you even leave the office, they can tell you, nope, you don't have strep throat. You got, a cold, and you have dripping in your throat from your sinuses, but it's not bacterial infection. There's no need for you to go on an antibiotic. In fact, it really wouldn't be beneficial for you at all. Rest, fluids, go home, call me in the morning. Or they can say, yep, you got strep throat, here's your Z-Pak, good luck. We are just now beginning to get a SNAP test for COVID-19, and that is potentially going to be a real game changer. Hopefully we'll be able to get things more open by being able to do fast 15-minute COVID-19 tests rather than the PCR test that takes several hours, and only specialized institutions are going to have PCR machines. COVID antibody tests you can do at home, you can do in the doctor's office, and before you even leave, you get the information. Lastly, one of the most modern tools of identifying bacteria is through DNA sequencing. But again, this requires a bit of specialized technology. We do it here, but it's not something that you would typically do at a hospital. So there are sequences within organisms that are referred to as a barcode, kind of like at the grocery store. It's not really a barcode, but it is a DNA sequence that is specific to a specific organism. And so you sequence a little piece, and you're able to identify what the organism is based on that DNA. It's not foolproof. There are mutations and things, but it actually works really quite well. All right. So this is our ultimate goal for identifying individual bacteria using sort of standard lab tools. And it is referred to as a dichotomous key. And so we can think of this as a whole list of questions, a series of yes-no questions that are going to lead you down a specific pathway. And it's a little bit like if somebody else has a destination in mind for you, but you don't know where it is, and you don't know where you're going, you're just told, get on the road, and you're going to just listen to your GPS. And at each intersection it says, go left or go right. And if we want to get you, and in fact this is kind of how map systems and modern GPS map direction finding works, is that you want to go to Urbana. And you have a whole group of places you could go to, or maybe let's go ahead and get a little bit further away. You want to go to Chicago. You could go to Chicago. You could also go to Springfield. You could go to Urbana. Heck, you go to St. Louis. At each point, you start and you begin making decisions about which way you're going to go. And, I mean, you personally don't do this, but this is what the algorithm that is identifying where you're going is doing. And so, it's a series of yes-no questions that are leading you to an ultimate goal. And just as an example, we've got step one, where you have taken bacteria, put it on a slide, and you're looking at a Gram stain. This is a stain that is going to darken and pick up stain whether the bacteria has a peptidoglycan coat or not. And if it's positive, if it's Gram positive, then you would go to step 2. If it's Gram negative, you go to step 6. All right, so let's just do that. Let's just say it's Gram negative. You go to step 6, pretty easy here, and now it's asking you to look in the microscope and say, do they have at least one flagella visible? If they do, you go to step 7. If they don't have any flagella visible, then you're at the end of the road. It is Escherichia coli. It's E. coli. So by doing this, you can work your way through a series of questions and end up at your destination, end up with the understanding and the knowledge of what this bacteria is. So we're going to stop here for today, and tomorrow pick up on some of the tools that are used for this and begin integrating a little bit of information from the lab as well. And that's what I have to say. So as we're kind of progressing here, I'm going to start thinking about how I can get you sort of a quiz that you can think about how well you're keeping up, and we'll see how things are going. I have to say, though, that I'm still trying to figure out how we're going to do the exams, and I think we're looking at doing an exam Monday, a week from this Monday. But we're a ways from that. Don't worry about that. I'm here to take care of you. We're going to make sure that we get through this. That's what I have to say for now. This is Dr. G, signing out.